Hello everyone, my name is Darshni Satimurthy from DBT3E. My metric number is 23 DBT20F1025. Today I am going to do a DNA extraction experiment. Let's start to do. In this experiment, you need to isolate some DNA from a human test subject. Why, you may ask, do you need human DNA? Because DNA contains the instruction needed for an organism to develop, survive and reproduce. To carry out these functions, DNA sequence must be converted into message that can be used to produce protein, which are the complex molecules that do most of the work in our bodies. Scientists isolate DNA for a variety of reasons, some of which include genetic testing, body identification, analysis of forensic evidence. DNA extraction is typically the first step in a longer laboratory process. DNA extraction is an important part of that process because the DNA first needs to be purified away from protein and other cellular contaminants. We need cells because that's where the DNA is. Inside almost every cell in our bodies is a nucleus, and inside each nucleus is about 2 meters of DNA. So, where do we begin? First, we need to collect some cells from our test subject. The skin on the inside of our mouth loses thousands of cells every day. These are the cells we are after. Let's get started. The steps you will follow to purify DNA from a chick swab are shown below. There are four basic steps of DNA extraction that are consistent across all the possible DNA purification. The first step is collect chick cells. The second step is burst cells open to release DNA. The third one is separate DNA from protein and debris. And the final one is isolate concentrate DNA. Here are the materials and equipment you will need to purify DNA from chick cells. Drag the swab over to the inside of the test subject mouth to collect a sample. Next, you will place the swab into an Eppendorf tube. Look closely at the swab. Note that it is covered with hundreds of tiny chick cells. Inside each chick cell is a nucleus and inside the nucleus is DNA. Place the tube into the warm water bath. The lysis solution you just added contained two important ingredients, detergent and enzyme called proteinase. DNA extraction is a method to purify DNA by using physical or chemical methods from a sample separating DNA from cell membrane, protein and other cellular components. The DNA is still wrapped very tightly around the proteins called histone, and the proteins cut apart the histone to free the DNA. The cells have stayed in the warm water bath long enough for the DNA to be freed from the cells, and we have removed the swab from the tube. Add some concentrate salt solution to your tube. The salt causes protein and other cellular debris to clump together. Place the tube into the microcentrifuge. In order to balance the centrifuge, a tube containing water in place opposite your tube. Click the lid to close the microcentrifuge and turn it on. Inside the centrifuge, the tube spins around at high speed. The heavy clump of protein and cellular debris to sink to the bottom of the tube, while the strands of DNA remain distributed through the liquid. Use the micropipette to carefully to remove the top liquid, which contains DNA, and place it into a clean tube. The protein and other cellular debris stay behind. Add some isopropyl alcohol to the tube. Inverting the tube several times, mix the isopropyl alcohol into your DNA solution because DNA is not soluble, does not stay dissolved in isopropyl alcohol. it come out of solution. Now can see the clump DNA with our naked eyes. Place the tube into the microcentrifuge and click the lid to close it and turn it on. Once the liquid is removed and the DNA is allowed to dry, you can redissolve in it the solution of your choice. DNA extraction is a routine procedure used to isolate DNA from the nucleus of cells. When an ice-cold alcohol is added to a solution of DNA, the DNA precipitates out of solution. If there is enough DNA in the solution, I will see a stringy white mass. The objective of this experiment, the DNA will be extracted using the basic biochemical technique for isolating, purifying and digesting DNA molecules. That's all from me. Thank you.